In recombinant protein production, researchers often face issues like insolubility or lack of expression. Tagging technologies help, but choosing the right tag can be tricky. Moreover, vector preparation, cloning, and testing can be time and resource-intensive. The EXIC ProGen Protein Expression Optimization Kit simplifies this process and improves solubility of difficult-to-express proteins. It includes 6 pre-selected tags for exploring optimal expression conditions across diverse targets. Tags can be added directly via PCR, eliminating the need for cloning or vector construction. The resulting genes are ready to use in automated cell-free expression systems to generate soluble proteins. After protease treatment, tags can be removed to isolate high-purity proteins for characterization. Streamline your workflow and optimize expression conditions efficiently with EXIC ProGen kits.